Hello everyone, myself Anuradha from Msc-2ndYear RA College Washim, and today we will see seminar on the topic Biosensors. Contents. In this topic we will see firstly the introduction about the biosensor, then definition of the biosensor, principles of the biosensor, advantages, some types of the biosensor, applications of the biosensor, references, etc. Biosensor. Firstly, introduction. Biosensor is an analytical device which converts a biological response into the electrical signal. It detects, records, and transmits information regarding a physiological change or a process. It detects the presence and the concentration of specific substance in any test solution. Now, the discovery of the biosensors. The father of the biosensor is Leland C. Clark. He invent a Clark oxygen electrode, a pivotal device that allows real-time monitoring of patient blood oxygen level and has made surgery safer and more successful for millions around the world. Now, the definition of the biosensor. In the definition, we will see the two types of definition of the biosensor. Firstly, the old definition and then the current definition. The old definition of the biosensor is that biosensor is a self-contained integrated device that is capable for providing specific qualitative or semi-quantitative analytical information using a biological recognition element which is in direct spatial contact with a transduction element. Now, the current definition of biosensor is that a sensor that integrates a biological element with a physiochemical transducer to produce an electrical signal proportional to a single analyte which is then conveyed to a detector. Now, the principles of the biosensor. Principles of the biosensor involves a three element. First, biological recognition element which highly specific toward a biological material analyte produces. Second, transducer detect and transduces signal from biological target receptor molecule to electrical signal which is due to a reaction occur. Third, after transduction signal from biological to the electrical signal where its amplification is necessary and takes place and read out in the detector after processing the values for are displayed for monitor and controlling the system. Now, the advantages of the biosensor. There are the different advantages of the biosensors. Firstly, it is highly specific. It is independent of the factor like stirring, pH, etc. Biosensors has a linear response. It is tiny and biocompatible. Biosensors are easy to use, durable. Biosensors require only a small sample volume. Biosensors are rapid, accurate, stable and sterilizable. Types of the biosensor. There are the different types of the biosensors such as calorimetric biosensor, glucose biosensors, optical biosensors, electrochemical biosensors and some others. The electrochemical biosensors can be classified into three types. Firstly, amperometric biosensor, conductometric biosensor and potentiometric biosensor. Now we will see the first type of the biosensor, calorimetric biosensor. Many enzyme catalyzed reactions are exothermic, generating heat which may be used as a basis for measuring the rate of the reaction and hence the analyte concentration. The advantage of this type of biosensor is that it is insensitive to the optical and the electrochemical properties of a sample. This type of biosensor is also called as a thermal detector biosensor. An example is use of glucose oxidase for determination of the glucose. Now, glucose biosensor. Glucose react with glucose oxidase to form a gluconic acid. 2 electrons and 2 protons are also produced. Glucose mediator react with surrounding oxygen to form hydrogen peroxide, that is H2O2, and glucose oxidase. Now, glucose oxidase can react with a more glucose. Higher the glucose concentration, the higher the oxygen consumption. Glucose content can be detected by Pretty electrode, which is as shown in the following diagram. Now, third type, optical biosensor. This involves determining changes in light absorption between the reactant and products of the reaction, of measuring the light output by luminescent process. A most promising biosensor involved luminescence uses firefly enzyme luciferase for the detection of bacteria in a food and or a clinical sample. The optical biosensors is as shown in the diagrammatic representation. Now, the electrochemical biosensor. Many chemical reactions produce or consume ions or electrons, which is turn cause some change in electrical properties of a solution, which can be sensed out and used as measuring parameter. The electrochemical biosensor can be further classified as amperometric biosensor, conductometric biosensor, and the potentiometric biosensors. Amperometry for applied current. The movement of electron in the redox reaction is detected when the potential is applied between the two electrodes. Conductometric is for the impedance, and potentiometric for voltage change in distribution of charge is detected by using the ion selective electrode such as the pH parameters. Now, amperometric biosensors. Amperometric biosensors are function by production of current when a potential is applied between the two electrodes, the magnitude of current being proportional to substrate concentration. These biosensors use to measure the redox reaction. The amperometric biosensor diagrammatic representation is as follows. Now, the potentiometric biosensors. These biosensors use ion selective electrode to convert the biological reaction into the electrical signals. Many reactions generate or use H positive ion, which is detected and measured by the biosensor. The urea biosensor is the example of the potentiometric biosensors. Now, the last part of the today's seminar, that is the applications of the biosensors. There are the many applications of the biosensors. Firstly, it is applicable in the food analysis. It is applicable in the study of biomolecules and their interaction. It is applicable in drug development, crime detection. It is also applicable in the medical diagnosis, both in clinical and the laboratory uses. It is applicable in the environmental field monitoring, quality control, industrial process control. It is applicable in the detection system of biological warfare agent, applicable in manufacturing of pharmaceutical and replacement organs. And the last is for applicable for the detection if diseases various proteins are sensed out in our body fluid. These are the some references. Thank you.